Okay. Good morning, everyone. This is our presentation, the campus learning presentations, and Caleb is going to get started with us for you guys.
Hello, everyone. My name is Caleb Lau. I'm majoring in biomedical science, and I'm currently a sophomore.
Okay, so my name is Joel Pasillas Gonzalez. I have an interest in genetics, and I am from Mexico.
Hi, my name is Ruchira Mirji. I'm majoring in bio with concentration in genetics, cell and developmental bio. And my fun fact is that my birthday is on Christmas.
Hi guys, my name is Mina Abdullah. I'm majoring in biomedical science. And a fun fact is that I broke the same wrist twice.
My name is Christina, and I'm also majoring in biological sciences with a concentration in genetics, and I'm from Romania. Okay, so when looking at our research interests and the experience that we've gained, the main reason why most of us decided to do this was because we wanted to gain research experience, things like pipetting, knowing how to load a gel to help us in the future when we start our own, start to work in our own labs, and even just in genetics lately in lecture, it's been helping a lot with what we've been doing all semester.
So for intro, the starting question that we had was, what does running PCR and gel electrophoresis on a DNA sample tell us about the Fc gamma and the APOA2 gene? And so our hypothesis was, if we replicate our extracted DNA from our saliva, then it will help us get a better understanding and detailed analysis of the Fc gamma and the APOA2 genes. And what we predicted was that we would either see wells in the H and R columns to be filled around the 500 base pairs in the first row, because that was what we used as the control group. And also either the H or R wells, or both of them, should be filled in order to determine if the individual was heterozygous, homozygous for histidine, or homozygous for arginine. And for the APOA2 gene, we should find that an individual is either homozygous for T or homozygous for C. And so the objectives of this lab were that was to analyze the gel electrophoresis data and determine the genotypes for the individual and find whether an individual is at a higher risk for obesity using the gel electrophoresis results of the APOA2 gene. And some background information was that the APOA2 is one of the most common proteins in high density lipoproteins.
Okay, so in order to conduct this lab, we were instructed to complete the procedure in several parts, starting off with extracting DNA. We extracted DNA using the extraction kit and collected around 1.5 ml of the saliva sample. And after the collection of saliva, several buffer solutions, such as the phosphate buffered saline, were added in order to equalize the pH of our solutions, which allows a higher yield solution. And the DNA sample is then mixed with protease, which breaks down any contaminating proteins into amino acids, whereas the buffer disrupts and breaks down. In order for the protease to break down unwanted proteins, the sample is then incubated at 56 degrees for around 10 minutes. And this allows the the solution is then added to a spin column and spun for 2 minutes in order to bind, and this allows the DNA to absorb to the silica membrane. The solution is then added to 2 buffers, and then we complete the process by eluting from the spin column. And for this PCR, the PCR contains of 3 main steps: denaturation, primer annealing, and elongation. And we start off with the hot start or denaturation at a temperature of 95 degrees for 5 to 10 minutes. The reaction mixture is heated for 30 seconds. And for primer annealing, the reaction temperature is reduced to 45 to 69 degrees Celsius for around 30 to 60 seconds, which allows the primers to bind the sequences. And next would be extension or elongation, and it's raised to 72 degrees Celsius. And this allows polymerase synthesis. We end the PCR with final extension for around 5 to 10 minutes, and the entire PCR process must be completed for around 30 to 35 cycles.
Okay, so looking at the results for the FCG gene, so we are going to look at so for the first line, that's the control line. So it just gives us a basis on what we should be looking at in terms of the size of the DNA. And so the second line is what we're going to be looking at. So in the left-hand column, this is the histidine, and then the right-hand column is for the arginine. So if an individual, like in this case, for individual 78, since they have both bands for both H and R, they are going to be heterozygous. And this means that they are in between two extremes. Therefore, they have a equal chance of infection. And then in this case with individual 96, we see the same thing where there's the light band under the control line for both H and R. And so this also shows that this individual is heterozygous for this gene. Okay, and then here we have an individual who has a band for the histidine, but not the arginine band. So this shows that they are homozygous for histidine, which means that they are more likely to be infected. Sorry, so if you're homozygous for histidine, you have a higher likelihood of being reinfected with dengue. And so this is one of the higher extremes. And then looking at individual 121 and 128. So individual 128 is going to be homozygous for histidine. And then individual 128 is going to be heterozygous.
Okay, and now we're going to be looking at the APOA2 gene. So this one was a little different because this one was split into undigested and digested. And so you can see it follows the same general principles in that if the fragment of DNA is smaller, it'll run through the gel faster. So in 121, 96, and 78, we all see that in the digested column, they all have a band that is between the 200 to 300, which means that they're homozygous, whereas in the 128 well, we see that she has two bands there, which means that she's heterozygous, and that's seen in the grand scheme of the likelihood of being obese in the future in terms of cholesterol. And then here on the next slide, we see just a summary of the data we've presented for both the FCG and the APOA2 gene.
Alrighty, and for discussion, so we did prove our hypothesis. We were able to take a closer look by running these samples through the gel and taking a look at both of these genes. In conclusion, so for the individuals that were homozygous for histidine at the single nucleotide polymorphisms for the FCYR2A gene, those individuals have a higher binding affinity to that immune complex, so they have a higher likelihood of being reinfected with dengue. And for the second project, for individuals who were homozygous for C, for the APOA2 gene, those individuals are at higher risk for developing obesity through the low HDL levels and food intake. And implications for this would be, so in terms of dengue, if a population is mostly comprised of homozygous histidine, it's gonna be a bigger outbreak, and officials have to account for that. And as for the second project, individuals with homozygous for cytosine and APOA2 gene should just be aware of the elevated risk of developing heart disease and making some lifestyle choices to address those and lower that risk of heart disease. So, any questions?